Dr. Abreu is the Associate Director of the Division of Neurogenetics and the Director of Lysosomal Storage Disease Program at NYU Langone Health. He also holds a position as an Assistant Professor in the Department of Neurology at the NYU Grossman School of Medicine. In addition to his clinical and academic roles, Dr. Abreu serves on the Patient Outreach Committee for the American Society of Gene and Cell Therapy. Thank you so much for being here, Dr. Abreu.
Thank you very much for having me, Calliope, in the APBD RF, and also for the candid and thought-provoking conversation by caregivers and people living with APBD. Most recently, certainly touched deeply. Would like to start with PhD candidate Rihanna Young from Newcastle University to share her exciting work.
So yeah, I'm Rihanna, and I'm a PhD student in White U's lab in Newcastle. And I'm excited to talk about my project, which is to identify and characterize inhibitors of glycogen synthase 1, a therapeutic target for glycogen storage diseases. And yeah, thank you very much for having me. So there was quite a lot of background covered, especially yesterday, about glycogen storage diseases and glycogen synthase as a target. So just to reiterate, the common hallmark of APBD, Pompe and Lafora is aberrant glycogen accumulation in affected tissues, which cause a variety of symptoms depending on the tissue affected. And glycogen synthase is a therapeutic target for this group of diseases, as it catalyzes the rate-limiting step in glycogen synthesis, which is the elongation of glycogen chains. So the rationale for this enzyme as a target is if we can reduce the amount of glycogen being synthesized, we reduce the amount that's left available to accumulate in affected tissues. And we did hear some of the studies throughout the last few days, but yeah, this rationale has been validated by some animal and cellular models of disease. So the strategy and workflow for my project is to utilize DNA encoded library screening as an initial hit finding mission, followed by machine learning to analyze the DEL data, and then validate hits using biophysical and activity assays, and then also do a structural validation as well to structurally validate hits that are found. So the main bulk of my work so far has been looking at DEL screening. And this is a screening method where compounds are synthesized from different building blocks, and each building block has a DNA tag on it that acts as a barcode. And there's repeated splitting and pooling of these building blocks, followed by the addition of the appropriate tag. And this allows synthesis of quite diverse libraries, but they're all present in a single solution. And this can be tested against an immobilized target such as GYS, and binding compounds can be identified by sequencing the barcodes. So in essence, it allows millions of compounds to be screened in tandem, which is in quite stark contrast to the one compound per well approach typically used in high throughput screening. So yeah, just to shout out to my collaborators, I've been working with Mike Waring's group at Newcastle, and also more recently with Jörg Schurman's group in Zurich, who are both setting up their own DEL screening platforms. This is some example data from my DEL screening. With the DEL analysis, what we do is break the compounds back up into their individual building blocks. And a hit can be indicated by either large counts of a single compound or by the presence of the same building block in multiple compounds, because during the synthesis, they're using the same building block sort of over and over again. So in this example here on the left, I'm not sure if you can see my cursor, but we can see this pane here in blue, looks like a sheet, and that's due to an enrichment in the position 3, building block 3. And then we can also see enrichment again in building block 1 and building block 2 within this building block 3 pane. I was really excited about this result initially, but further testing showed that unfortunately this was a false positive for these hits, which was disappointing. But since then, I have continued to work on the screening protocol, and I have reduced this background noise so these hits won't come up in further screens. I've also been looking to improve the immobilization of my protein, and the chemists are continuing to generate new libraries to continue this. And yeah, Dell does generate quite large data sets. I think at the moment the chemists are working with a library around 3 million compounds. So you can get quite a lot of data out of the screen, which would be compatible with machine learning analysis, which we're quite excited about as well. So moving away from the DEL and onto some basic activity assays, I have been optimizing this UDP glow luminescence assay using a control inhibitor established in literature called analog 23. And yeah, I use this to optimize a commercial assay, and it's been working quite well. I've got quite a nice profile here for analog 23. So moving forward, I'm going to use this compound to develop a binding assay and also to optimize conditions for structural determination. I'm using cryo-EM. So this has been quite a useful compound for me. Following on from that assay work, I was also able to characterize some GYS1 inhibitors that were provided. They were originally discovered by Roche, and then in collaboration with Berge Manassian at Southwestern, he provided these compounds and mediated a collaboration between us and Patrick Gunning's group at the University of Toronto. So I was able to sort of characterize the activity of these inhibitors, and Patrick's group have been developing a number of small molecule derivatives that I've also characterized in the same way, and they're also developing PROTACs, which will reduce the impact of GYS by marking it for degradation. So that's quite an exciting prospect. So yeah, I'm looking forward to continuing to work with this group and hopefully characterizing more compounds and PROTACs, and yeah, building on this further once I have my binding assay in place. So site-specific targeting and cryo-EM. So structurally validating and characterizing inhibitors is quite an important component of the workflow because we are looking to target specific sites. So previous work within my group has used cryo-EM to determine the GYS1 structure and also give insights into the regulation mechanisms of the enzyme. So this is a map that I generated, and there are 2 sites of interest. Of course, we're interested in targeting the active site, but we're also quite interested in targeting this allosteric site here at the dimer interface. And the rationale here is that we could reduce GYS activity by disrupting its interaction with its allosteric activator, glucose 6-phosphate. So yes, I was able to generate this map, but this is without any compounds or inhibitors. And I've recently started preparing samples and I've recently screened some grids for cryo-EM to try and determine the structure of GYS and its interaction with inhibitor analog 23, my control compound. And I hope that this will put me in good stead and help me optimize conditions and sample preparation for looking at other inhibitors in the future. So just to summarize, The individual components of the proposed workflow are starting to take form. I am working to characterize some established inhibitors while simultaneously optimizing the DEL screen to try and identify novel binders. And my future work will include setting up this binding assay and optimizing conditions for cryo-EM. So thank you very much for listening. And of course, I've pointed out to quite a few collaborators. Collaboration has been a really important part of this project. So thank you so much to everyone who has worked with me, and also to APPDRF and the $1 Million Bike Ride, who have supported parts of this project, especially the DEL. So yeah, thank you very much.
Thank you very much, Rihanna. We'll take questions in the chat as at the end of our panelist presentations, and we'll move on to Dr. Benjamin Kuchenauer, my apologies, resident physician at Duke within the Division of Medical Genetics.
All right, thank you so much for to the organizers for inviting me, and to the it's been a really great conference to learn a lot about the exciting work going on in APBD. And so today I'm excited to talk about polyglucosin and what I'm calling the proximal disorders of glycogen synthesis. I think there's 3 rare genetic conditions that can all benefit from one another, and I'll try and go through some of that with you now. So for the overview, first I want to talk about in this brief talk the concept of the proximal disorders of glycogen synthesis, which I'm including glycogen storage disease type 15, which is related to glycogenin, GSD0A and 0B, which is related to loss of function of glycogen synthase, of course GSD4 or APBD, related to loss of function of glycogen branching enzyme. Rihanna's talk was excellent, and so we'll again review some advances from her lab and other labs around the world that have been shining light into the structural, new structural knowledge of all of these proteins. And then we'll discuss some possible therapeutic avenues, though Rihanna just gave an excellent overview of one of the most exciting, which is GIS1 inhibition. So these are the disorders that so I'm a physician scientist and medical geneticist, and I am fortunate to work with Dr. Kishnani and Dr. Koch on a number of these rare glycogen storage disorders, so I'm able to see all these in clinic. And these, I think, go together mechanistically. So GSD15 is related to glycogenin. As we said, GSD0 is glycogen synthase, and GSD4 or APPD, of course, is the branching enzyme. These conditions are all autosomal recessive, and they're due to loss of function, so losing either glycogenin, glycogen synthase, or glycogen branching enzyme. Prior to our work, there was no liver phenotype for GSD15, though we'll discuss some new clinical findings there. There has been known to be liver involvement in glycogen synthase with ketotic hypoglycemia, which is related to the inability to form glycogen in the liver. And of course, GSD4 has variable liver involvement, which can be quite severe. The skeletal muscle involvement has been described for all of these disorders. There's a sort of matching disorder for APPD, which is called PGBM2 or polyglucosin body myopathy type 2, which has a lot of the skeletal muscle and skeletal muscle weakness features that are very similar between APPD and this disorder, though APPD has more of a CNS and PNS involvement than this disorder. And they also all have cardiomyopathy that has been linked, whether it's arrhythmia or deposition of this polyglucosin, this poorly formed glycogen granule that can cause issues with the heart. These are a couple of papers I wanted to highlight that have come out from different labs, including some of the co-authors that Rihanna was just discussing, including from her PhD lab, Dr. Yu, that's really done excellent work for the last decade or longer on these disorders, and I really appreciate the work that they've been doing to help us to understand how these proteins work. And this is from a paper from Mar et al. That just kind of goes through why I think these 3 proteins should, in our head, be linked, and why I think advances in the treatment of one or all these disorders are useful for all three communities. So glycogenin is a small protein which has this tyrosine residue here that will grab onto the first residue of glucose as you're building a glycogen chain. So here you can see that after the arrow, the transfer of this orange glucose molecule onto the cartoon of glycogenin. Another UDP glucose comes and continues the growing chain, and this continues on for about 6 to 10 residues. Glycogenin is in a complex with glycogen synthase, and so this is the same tetramer that we saw in Brianna's talk, a cartoon version of it. And once the glycogenin has enough glucose residues, it will pass it into glycogen synthase, which then synthesizes that linear chain of glucose into a longer string that our hero or villain for APBD, the glycogen branching enzyme, then branches all of that linear glucose polymer into the nice glycogen. What happens in APBD is you're missing glycogen branching enzyme, and so you're not able to have this nice kind of circular branched glycogen, and so it becomes very long and linear, and it's insoluble. And that's what builds up in tissues and causes the issues. This is just another cartoon from the cryo-EM that lets us look closer, and this dovetails well with Brianna's work to look for inhibitors of glycogen synthase. And now, with knowledge of how glycogen synthase and glycogen interact, we have even a better opportunity to really understand what those targets are and how to block them. And so, what came from this study and from another study that came out in 2022 was this autoinhibited state with phosphorylation. And so now we have more understanding of these various states of the glycogen and glycogen synthase complex, and which gives us the opportunity to design new therapeutics to push this complex in one direction or the other, depending on what our therapeutic goal is. This is from our paper that's focused more on glycogen storage disease type 15 that will be coming out soon, where we've identified a set of patients that have missense variants and glycogenin that have liver involvement, to kind of further fill in the picture and show that all these 3 disorders have similar clinical phenotype. But I apologize, kind of a busy figure, but we'll start at the top just so that you kind of understand why I feel like these disorders all should be in our mind together. At the top, we have UDP glucose, which is bound not necessarily separately by glycogenin and glycogen synthase, but in fact by this glycogen and glycogen synthase complex. So on the outside here are 4 monomers of glycogenin. On the inside are these red, purple shades of glycogen synthase. They produce that intermediate glucose polymer, which then glycogen branching enzyme will turn into normal branch glycogen. So that's the non-diseased pathway. For glycogen storage disease type 15, when glycogenin is not working well, the missense variants are actually worse than the null variants. So that in the missense variants, those patients present early with severe cardiac disease, at least for a subset of the missense variants. And it's because the glycogenin is still there making a complex, but it's blocking the site for glucose to diffuse in and turn into glycogen. So glycogen branching enzyme is normal, however, you still end up with polyglucosan. If you're completely missing glycogenin, glycogen synthase can still work, just not as effectively. And for reasons that are not completely clear, you still form an abnormal intermediate polymer that becomes polyglucosan. And this is the basis of the polyglucosan body myopathy type 2, which is a very similar disorder to APBD. The next 2 disorders are GSD0A and B, and they depend upon whether that particular gene is expressed in the liver or in the heart and the muscle, and the symptoms correspond with either liver disease or cardiac and skeletal muscle disease. In these disorders, we see decreased branch glycogen, though there is at least some animal model data that there may be some polyglucosan in this setting, so further work is needed to kind of understand if there is if the glycogen that is made is normal or abnormal. And then, of course, in the setting of GSD4 or APBD, this complex is all normal. We're just missing GBE. And there's variability with kind of the classic, what would be called GSD4, which is the early onset form. Typically have full loss of GBE, so null variants or deletions, whereas the APBD tends to be more related to specific missense variants, that the protein is there, but you're able to branch, but not as effectively. And that's why it's a very slow onset of disease later in life. And so as we were discussing in the prior talk, the main goal then in APBD, the therapeutic options would either be to use something like a gene therapy or an mRNA therapy or some other therapy to bring back normal levels, or at least some level of glycogen branching enzyme. Another option would be to inhibit glycogen synthase. which I believe has been discussed at length in the conference. And so I think these are all, exciting avenues to look at. And then, of course, another avenue would be if there are possible ways to kind of melt away this polyglucosan that's been building up. And so now with our work in GSD15, we can sort of fill out this table so that all three of these disorders, which I argue are related in important ways, and each will kind of bolster the effect of the research impact of the other. And so we're able to now add the liver effect there too, and we're hoping to have that out by the end of the year. This was just a plug, but now we just went through it and a full talk about why we would want a glycogen synthase inhibitor. There are, in addition to the efforts by Rihanna, there's been another group in the US that's made it to clinical trials with this compound. It's very exciting. That does do a very good job of inhibiting DIS1, which I think I'm very excited to see. We'll have, there's also other groups looking at RNAi, so a number of ways of knocking down DIS1, which could be beneficial not only for Pompe disease, but of course for APBD, which I think is a very exciting avenue for therapy. So I'll kind of wrap up there and just go through some of the future directions. So I'm excited to be done with training finally in 3 months, and I'll be starting my lab early next year. And so I'm excited to start to dive in and work on some of these questions and hope that we can move towards really the first therapeutic option for APBD, first targeted therapy. But some open questions that still exist, what are there subtypes of polyglucosan, this misformed glycogen, and do those differences in the way that polyglucosan forms, do they matter, and can we target them different? So I think there's still further work to be done there. Another question is, in GSD0, is there also polyglucosan and that we can look at with a number of models? And then we've kind of discussed, but there are a number of therapeutic avenues that I think are very exciting for all of these disorders, but we'll focus on for APBD. So as we mentioned, one option is to restore the normal function of the missing protein, whether that's gene therapy, which is an obvious path, but challenging to do in practice. Whether there are antisense oligonucleotides that could correct splicing or potentially increase gene expression, depending on the particular variant within patients, is we've been moving as a field more towards N-of-1 therapies, and I think this would be a possibility depending on particular individual patients and their variants, and whether that would be a feasible option. We also can look at decreasing the production of polyglucosan, and that's the idea of modulating that glycogen synthase glycogen complex activity through small molecules, RNAi, other avenues to do that. And I think that is really a maturing field, which is exciting. And lastly, there's been a lot of work, and I believe that the group from Israel already presented on this work either today or in prior meetings about removing that polyglucosan by targeting some of the lysosomal proteins, which is an exciting avenue as well. So I want to say thank you to my postdoc lab. This work has primarily been done in my with my mentors, Dr. Kishnani, in collaboration with Rebecca Koch, who's brilliant, wonderful to work with, and Deek Shapali in our GSD lab at Duke. Thank you so much again for inviting me, and I'm happy to take a look at some questions in the chat.
Thank you. Next, we'll have doctoral candidate Sophia Desasare from UT Southwestern Medical Center. Thank you.
Good afternoon, everyone. As introduced, my name is Sophie. I just began my second year as a graduate student in the NICHQ lab at UT Southwestern. Today, I'll be sharing part of my project that focuses on developing gene therapies for APBD. As you've heard in previous talks, polyglucosan bodies are one of the main pathologies seen in APBD. These are deposits of insoluble glycogen that accumulate and disrupt the normal function of the nervous system. They are caused by defects in glycogen branching enzyme, or GBE, and therefore glycogen lacks the normal amount of branch points it would have compared to a healthy patient with normal glycogen. What's interesting about these deposits of insoluble glycogen is that they actually resemble another polysaccharide called starch. And starch is another glucose storage macromolecule that is naturally supposed to consist of these longer linear chains, as shown here. It's made up of components like amylose and amylopectin, which you can see are quite linear. While humans do not store glucose in this structure, we store it in glycogen. We do produce an enzyme that is capable of breaking it down. We produce amylase, mainly in saliva and the pancreas, to break down dietary starch. It has been shown that amylase is also able to break down the PGBs seen in diseases like APBD. For example, the Gentry group demonstrated that delivering a bioengineered amylase to diseased mice via ICV was able to reduce PGBs in the brain, shown here. This treatment is called BAL0417, and it's the protein therapeutic that we based our project off of. We aim to modify this therapeutic and turn it into a gene therapy for a more permanent treatment solution. By bioengineering a protein construct to have cross-correcting abilities, it is then able to translocate from treated cells into untreated cells and increase the treatment efficiency as it spreads. So to make a protein be able to cross-correct, you need to add two components, a secretion signal that allows the protein to leave the cell it was delivered to, and secondly, a cell-penetrating peptide, or a CPP, that allows the protein to be taken up into untreated neighboring cells. So cross-correction can be done to any protein, and we wanted to apply this idea to an amylase gene therapy. So we started with making a vectorized version of the protein treatment I previously showed from the Gentry group, that BAL0417, and we called this construct 1. The protein was bicistronic, so basically two components that come together into one and contained a Fab antibody fragment. We modified it to be a single chain and switched the Fab antibody to an even smaller antibody fragment called FB. The smaller fragment was either a mouse or a humanized version in order to compare which version would be more efficient. We also screened for an ideal secretion tag and found that human albumin was the most effective at secreting the construct. Finally, we tested two different CPPs to see which one would create the most uptake. Both are different variants of TAT, either a TAT-K or a cyclic TAT-K. And here's a table that details the different construct components. So again, construct 1 is the vectorized BAL0417. N designates the mouse version of an antibody fragment, and it has either a no CPP or one of the TAT variants, TAT-K or cyclic TAT. And H is the humanized version of the antibody, again, either with no CPP or one of the two TAT variants. So each of our constructs also contains a HYBIT tag. To explain a little bit more about what this is, HYBIT is a small portion of a fragmented luciferase system. When HYBIT binds to large bit, it produces a functional luciferase called nanobit that can be used for easy monitoring of our constructs. The HYBIT system is beneficial because it is a really small tag. It's only 11 amino acids, so it doesn't interfere with the normal function of the protein or its secretion from the cell. This assay can be adapted in many different ways. Here we use it to detect our constructs in lysate, in secretion assays of conditioned media, and in live cell detection for uptake. All we need to do when it comes time to read the assay is to have all three components present. So the two portions of the luciferase and the substrate, then we'll be able to generate a luminescent signal that corresponds to our protein levels. So I tested these constructs in an in vitro cross-correction assay. This assay involves transfecting the plasmids into wild-type HEC293 T cells, turning them into what we are calling producer cells. These producer cells will now secrete the constructs and act as if they were the corrected cells that were successfully targeted by a gene therapy. Then the next day, I add in receiver cells into the same dish, creating a co-culture. The receiver cells are almost like the wild-type HEC cells, except they contain the high bit or the large bit portion of nanobit inside the cell. So the idea here is that the secreted construct will leave the producer cell and be uptaken into the neighboring, essentially untreated cell, and where it will produce the functional luciferase to indicate successful uptake. This luminescence is measured and compared between the constructs. We found that all the constructs do show significant uptake, indicating successful cross-correction. And the values range from anywhere from 5 to 12-fold higher uptake signal when it's normalized to our GFP control. So from these constructs, we found that the mouse antibody tends to perform a bit better than the human antibody fragment. And therefore we selected the mouse antibody with the two different TAT variants to turn into a virus. So the previous constructs were in a backbone that were missing components that are necessary for AAV production. So we cloned the constructs into a new backbone to make single-strand AAV. This backbone contains a strong CDH promoter and a WPRE3 enhancer element, as well as all the necessary viral components like ITRs. We wanted to confirm that the construct still functions in this new backbone, so we performed the same secretion and uptake assays with the new plasmids. The viral vectors of the constructs do work, and even seem to outperform the previous versions of the construct. So you can see here in the graph that I designated the viral versions with a V and this little graphic. So now, since we confirmed that the new constructs are still able to produce the desired result and show promising secretion and uptake levels, these plasmids were sent to Baylor College of Medicine to be packaged into a virus for in vivo treatment. So we do have the virus ready now, and we're almost ready to start injecting the mice. I'm currently working on expanding our mouse line for experiments and practicing my ICV injection technique. We will be injecting the virus into P1 mice, so essentially the same location that's shown in the picture here, but much smaller, younger animals. Then we will age the mice to 3 months before staining with PASD and evaluating the insoluble glycogen content. Our rationale for testing the younger mice is to give the virus the best shot of working in this initial proof of concept tests. We just want to see that the virus can degrade the initial glycogen that begins to form deposits, similar to the aberrant glycogen shown here. And if we can see that our virus actually works in the brain and can degrade the insoluble glycogen that begins to form in vivo, then we will use it on older mice with more established deposits to test if it works on those bodies as well. So these injections should begin within the month, and I'm really excited to continue this work and be able to share the results. So with that, I'd like to thank you for listening to the presentation. Thank you to everyone in my lab and the Manassian lab for helping me with the virus production. And I guess I'll take any questions in the chat.
We'll start with the Q&A now for the remaining 7 minutes. Try to get through the questions that at least I've seen, and then any additional questions after the 3 o'clock changeover we'll address via the chat. So going back to Rihanna, you may have seen this, but the GIS1 immobilized in the same site in your assay. And then if you can follow up with the UDP glow question.
Yeah, of course. So for the activity assay, I don't need to immobilize the protein, so I don't use any immobilization for that. But when I'm hoping to develop an SBR binding assay, so I'll be immobilizing it using the same strep tag for that, so it will be kept consistent there. With the UDP glow assay, yes, it is a coupled assay, so there's another enzyme in the mix. So when I test a compound, I just test it, but I put everything, all the other reagents and substrates in other than GYS. So yeah, I that should sort of highlight any false positives there.
Thanks. Dr. Kalkenhofer, can you just talk a bit about your response?
Sure. Yeah, the question was a good question. So it was, how could loss of glycogen synthase, which is that middle portion, cause polyglucosan? Because polyglucosan, of course, in branching enzyme makes a lot of sense. There's no enzyme to branch. They have very nice names. And so my argument is that certainly full deletions of GIS1. So if there's no glycogen synthase around, I don't think that would cause polyglucosan. It would just cause no glycogen to be formed. But you could imagine instead a missense variant or a change in the active site of glycogen synthase that might be able to start some of the growth, but maybe having unusual or incorrect arrangement of glucose or incorporating other changes within the growing strand that could cause a polyglucosan-like or abnormal glycogen granule to be formed, which is all conjecture, but that was the question about possible mechanism.
Thank you. For Sophia, we had a previous question about outcome measures, or kind of when looking at mice, what is considered successful. So when you're starting to look at mice at that, I believe you said like 3-month mark or different time, do you have a target percentage or specific amount of glycogen reduction that you're aiming for? And if you were to think about treating older mice, if everything were to go as planned, what kinds of things might you consider in approaching the older mouse population?
Yeah, that's a good question. So I think for the first trials of these experiments, we just want to see the highest percentage possible of deposit reduction. I think there's also kind of a question about how much do we need to reduce the deposit formation to see an actual therapeutic effect. But I do think that we can start addressing that question if we're able to see an effect in just the proof of concept experiments. For your second question about how we would transition this into older mice, I'm not necessarily concerned about the functionality of the amylase in an older mouse, just because the structure of the polyglucosan bodies is the same regardless of if it's aberrant or if it's an actual just established deposit. So I do think it'll work to degrade it. I think it just kind of goes back to the question about dose and how much we need to deliver to see an effect.
Yes, from my end, Sophia, would you expect any kind of like off-target effects from secreted amylase outside of like what
I
you
think
want?
it's possible. We briefly mentioned looking at skeletal muscle to see if we're able to see a reduction in deposits of there as well. I think it's with especially with AAV9, like it's the strongest serotype for the brain, except it doesn't always stay in the brain. So I do know that it is able to kind of translocate to other more systemic locations in the body as well. And we'll definitely look at if it has an effect on the systemic bodies and the deposits we see in like skeletal muscle, for example.
Dr. Kochenoffer, I don't know if you want to talk about like at least like within the different different disorders you mentioned, just based upon the CNS and nerve involvement known in APBD, if you see CNS and nerve dysfunction in some of the other conditions.
Yeah, that's a great question. So in general, among those three, the peripheral nervous system and central nervous system involvement is the greatest and GSD4. I don't have a great explanation for that. But one lesson from comparing all three disorders is thinking about some of the treatments we're proposing, which I think are very reasonable. But like GIS1 inhibition, complete loss of GIS1 can lead to sudden cardiac death in children, and so I think we have to be very careful with thinking about getting the right amount of inhibition, and not too much inhibition can be dangerous. And so we can learn lessons from even as we're moving towards trials and with some of these different options, looking at other disorders that may already have some of those side effects. We already have rare genetic disease, but good data from what happens if you have complete loss of GIS1, complete loss of some of the other targets that we're trying to do kind of clever things with this therapy. And
thanks. We have one question that just came in. Sorry. Sophia, very nice work with the young mice. What level of resolution is there for showing how far the bystander effect may have gone?
I suppose. Well, could you repeat the last question?
Yeah. What level of resolution is there for showing how far the bystander effect may have gone?
I think we will mainly be able to look at the levels of soluble versus insoluble glycogen and staining to see like how much it's been reduced in the comparative tissues, and to see if we're noticing any bystander effect.
Thank you to the panelists very much for presenting your work.